Our next speaker is Dr. Vanessa Brait. Vanessa is a postdoctoral researcher at the Florey under the dementia research team led by Associate Professor Amy Brodman. Her research focuses on determining the mechanisms by which brain ischemia drives neurodegeneration and cognitive decline, and she's going to be speaking on middle cerebral artery occlusion does not produce hippocampal atrophy in wild-type mice unless directly infarcted. Vanessa.
Thank you.
Thank you.
So today I'm going to be speaking to you about stroke-inducing brain atrophy in mice, focusing mainly on the hippocampus. So as we all know, dementia and cognitive impairment are becoming increasingly recognised as major post-stroke sequelae. In fact, the frequency of dementia is 4 to 6 times higher following stroke than that in age-matched people without stroke. And up to 2/3 of stroke survivors develop some form of cognitive impairment. In the clinic, remote brain atrophy after ischemic stroke has been observed and has been associated with cognitive impairment. However, it is not known if these are a direct consequence of the stroke or of chronic midlife risk factors. And no studies have examined brain atrophy post-stroke in mice over long periods of time. However, some have found hippocampal atrophy. So this project aimed to examine the volume of the hippocampus and the cortex up to 48 weeks post-stroke, as well as to observe whether remote brain atrophy occurs in these areas. So we used 6-week-old male C57 black 6J mice, and we induced a 30-minute intraluminal filament-induced middle cerebral artery occlusion, or an MCAO, to model ischemic stroke. So very briefly, this is done by inserting a silicon-coated filament into the external carotid artery up through the internal carotid artery into the circle of Willis, where it blocks the middle cerebral artery at its origin. And during surgery, we are recording regional cerebral blood flow in the area perfused by the MCA with a laser Doppler. And this is a representative trace where we see a sharp drop in blood flow when the filament is inserted of approximately 80 to 95% of baseline. This is then maintained for 30 minutes, after which the filament is removed, and we induce reperfusion that returns to baseline within 10 minutes. So this is a longitudinal study where we assess mice up to 48 weeks post-stroke, and we use a 4.7 Tesla MRI for the brain volume assessment. So we first scan the mice before the surgery to get a baseline image. We then induce the stroke or the sham. We scan again at 3 days to get the infarct volume, and then we scan at 1 week, 4 weeks, 12, 24, 36, and 48 weeks post-stroke. At each of these time points, we produce both T2 weighted images, which are a structural scan, and we use this to manually delineate the hippocampus and the cortex. And we also produce various diffusion weighted images that assess white matter integrity. And Werda will be speaking about that in the next talk, but she'll be speaking about the rats in the study, not the mice. And I'll only be speaking about the T2 weighted images today. So now on to the results. I'm firstly just starting with some sensory motor tests. And although the ischemia of 30 minutes did produce a relatively mild ischemia, which we did on purpose just to try to make sure that the mice did survive the 48 weeks post-stroke, we do still produce significant motor impairments initially, and that are also maintained over the 48 weeks. In both the POL test, which evaluates motor skills and coordination, and also the adhesive removal test, which measures mouth and paw sensitivity and dexterity. So these are some representative T2 weighted images of a sham and a stroked mouse at each of the time points measured. I'll actually be showing you in the next slide the hippocampal volume, and then later the cortical volume measured. But when just looking at the images, we can't see any gross changes occurring in the shape or the size of either the hippocampus or the cortex at any of these time points in the sham-operated mice. However, in the stroked mice, clearly we see quite a different situation. So in the left is a normal-looking baseline scan. At 3 days post-stroke, we see this hypo-intense region representing the infarct volume, which encompasses, in this case, the cortex, the hippocampus, a little bit of the thalamus, as well as the striatum, although we can't see that in this image. We also see a midline shift due to a large amount of edema occurring in the ipsilateral hemisphere. At 1 week, the edema has mainly resolved. We see a relatively normal looking hippocampus and cortex. We do see a little bit of atrophy occurring here. By 4 weeks post-stroke, we definitely start to see atrophy occurring in both the hippocampus and the cortex. We see this expansion of the lateral ventricle, and we start to see a midline shift in the opposite direction to what we were seeing at day 3. At 12 weeks post-stroke, we see further atrophy in both the hippocampus and the cortex and more expansion of the lateral ventricle, and we see very similar images at 24, 36 and 48 weeks. So this is the hippocampal volume results. So at the top are just some representative images where you can see the manual delineation of the ipsilateral hippocampus in green and the contralateral hippocampus in red. So this is a sham operated mouse, a stroked mouse at the same level of the brain, and also the same stroked mouse but a more caudal image. And when looking at the graphs, we can clearly see atrophy occurring in the stroked mice, which are the red circles, compared to the sham operated mice. So we see a small difference at 1 week, more atrophy occurring at 4 weeks, further atrophy at 12 weeks, after which we see a plateau. And we don't see a difference between the sham or the stroked mice in the contralateral hippocampus. Now, interestingly, we do see an increase in the ipsilateral hippocampal volume in the sham-operated mice at 24-week time point that is then maintained until the 48 weeks. And we see a similar thing happening in the contralateral hippocampus of both the shams and the stroked mice. And I'll just refer back to that in the next slides. So we were interested in dissecting this atrophy that we're seeing further in order to understand whether it's due to a direct infarction or whether it could in fact be considered a remote atrophy. So we took only the stroked animals and subdivided them into those that had an infarcted hippocampus and those that did not. So the green squares were the stroked animals without an infarct in the hippocampus, and the yellow circles had an infarcted hippocampus. And it's very clear to see that we only see atrophy occurring in the hippocampus when the hippocampus was itself infarcted. Because this, we can see, is quite similar to what we saw in the sham-operated mice in the previous slide, with an increase at the 24-week time point that is maintained until the 48 weeks. We also see a similar thing happening here in both groups as well. So I've actually taken these 4 groups, as well as the right and left hippocampal volume of the sham-operated mice from the previous slide, and compared them onto one graph. And we can clearly see that there is in fact a very, very similar profile in the hippocampal volume in all of the groups, apart from the ipsilateral hippocampus of the stroked mice with an infarcted hippocampus, strongly suggesting that in wild-type mice, the stroke lesion itself does not produce brain volume changes remote to infarct-affected areas, at least not in the hippocampus. Now, in order to establish whether these reductions in hippocampal volume in the mice with an infarcted hippocampus were occurring across the entire hippocampus or only in specific regions, we divided the hippocampal segmentations into the dorsal and the ventral sections. Now, these are functionally distinct areas, with the dorsal hippocampus being mostly important for cognitive processes such as learning and memory, and the ventral hippocampus more important for emotions and motivation. So you can clearly see that both the dorsal, which is the purple section in the image, and the ventral ipsilateral hippocampus atrophy when there is an infarct in the hippocampus. And we don't see any changes in the contralateral hippocampus in either groups. And that's also very clear in this representative image, where you see the ipsilateral hippocampus, the dorsal in purple, the ventral in green, clearly atrophied when there's an infarct, but the contralateral, there's no difference between the 2 groups. So we also measured cortical volume in these animals as well. So again, representative images with the ipsilateral cortex in green, contralateral cortex in red. This dotted line is simply drawn at a landmark at the anterior commissure, which we chose simply because after that point it made it a little bit difficult to differentiate between subcortical and cortical regions. So this was chosen to make the volume measurements more consistent across brains and time points. So when looking at the graphs, again, we see significant atrophy in the ipsilateral cortex after a stroke compared to the shams, and we don't see a difference in the contralateral cortex. So just like we did for the hippocampus, we separated the stroked mice into those that had a cortical infarct and those that did not. However, in this case, the data isn't quite as nice or as clear because only one animal did not have a cortical infarct, which meant that the stroke in that case was quite small and was only striatal. But anyway, we can still, so I haven't been able to do any stats or anything on this, but we can still see that it does look potentially like there's only atrophy occurring in the animals that had a cortical infarct as well, because this is also very similar to what we saw in the previous slide, the shams. So just to remind you, so what we see here. So this also potentially suggests that in wild-type mice, the stroke lesion itself does not produce cortical volume changes remote to infarct-affected areas. However, we can't read too much into this given that there's only an N of 1. So in summary, we found significant atrophy in the ipsilateral hippocampus at 4, 12, 24, 36 and 48 weeks post-stroke, but only when the hippocampus was directly infarcted. We found a similar profile in the hippocampal volume of all mice, except those with an infarcted ipsilateral hippocampus, suggesting that the stroke lesion itself does not produce hippocampal volume changes remote to infarct-affected areas. Now, this is quite different to what is found in the clinic. However, what's been mentioned previously as well as in the Canvas study, brain volume is already compromised at the time of stroke, suggesting that chronic midlife disease processes are involved. And we have to remember that the mice used in this study were young, they had no comorbidities and no vascular risk factors. So the next step is to repeat this study with either hypertensive or type 2 diabetic or aged mice, and that'll be really interesting to see. So when the hippocampus was divided into the dorsal and the ventral sections, we found significant atrophy in both the ipsilateral, dorsal and ventral hippocampi in the stroked mice with a hippocampal volume infarct. We also found significant atrophy in the ipsilateral cortex from 4 weeks onwards, potentially only when the cortex was infarcted. And overall, these findings demonstrate novel temporal, hippocampal and cortical volume changes up to 48 weeks post-sham and post-stroke. Thank you very much.
Thank you, Vanessa.